important छ याद गर pedigree chart and the symbol used in the pedigree chart. And next one is the Mendel's law of inheritance. So we have to know about the three different Mendel's law. So this one is a kind of revision for you. DNA RNA त तिमीले पढिसकेछौ हैन प्लस 2 मा. So this is a kind of revision for you. So DNA is the genetic material of the inheritance, which was established by Watson and Crick in 1953. So just see this diagram. You can see the double helical structure. Can you see the double helical structure with string of the nucleotide? Now when you closely observe this part, this is known as the nucleotide. So a single nucleotide, it consists of a sugar molecule. It consists of the phosphate group, and next one is this one. This one is the nitrogen base. So whatever you are seeing, the A, C, T, G, those are known as a nitrogen base. So the sugar molecule, single sugar molecule, phosphate group, and the nitrogen base, this made a single nucleotide. So we have got the double helical structure, which are made of several unit of a nucleotide, several unit of the nucleotide, which are attached with one another. So this is the definition of the DNA. Now in a DNA, in the case of the sugar molecule, or in the name of the sugar molecule, we have the deoxyribose. But in the case of the RNA, in a sugar molecule, we have got the ribose. So that is the difference between the DNA and the RNA. As this consists of the deoxyribose, the name is known as a deoxyribonucleic acid. Now next one is DNA chemical structure. So you just see the nitrogen base. I've already told you about the 4 different bases which are present over there in the DNA. Now those are known as the purine and pyrimidine. So the purine is the double ring. So this is the double ring. Guanine and the adenine, those are known as the purine, and pyrimidine are thymine and cytosine. Now the guanine always pair with the cytosine, and the adenine always pair with the thymine. So this is the pairing of the DNA. Now the RNA, the full form of the RNA is the ribonucleic acid. Unlike that of the DNA, the RNA only consists of the single strand of the nucleotide. We have the single strand of the nucleotide. You can see the single strand of the nucleotide, which is synthesized from the DNA. And there is the presence of the longer of the nucleotide unit. Now the same thing, the nucleotide unit, it has got the sugar molecule, it has got the phosphate group, and it has got the nucleotide. It has got the nitrogen base. Now just see, in the case of the sugar molecule, there is the presence of the ribosome. And next one, in the case of the nitrogen base, nitrogen base, we have the guanine, cytosine, adenine, thymine is there in the case of the DNA. But in the case of the RNA, the thymine is replaced by the uracil. So cytosine is there, guanine is there, adenine, and the uracil is there. Now we have mainly the 3 different types of RNA: mRNA, tRNA, and rRNA. Now this one is known as the mRNA. Just say these are the difference between the mRNA, tRNA, and RNA. mRNA is a single and uncoiled strand. You can see this is a single and the uncoiled strand. And what's the main function of the mRNA? The main function of the mRNA is to produce the protein. The protein synthesis is the main function of the mRNA. Now next one is the tRNA. tRNA is this one. This is the tRNA. Now over here, the tRNA, you can see the single strand of the RNA is folding back on itself, and it is forming a kind of similar type of this structure. This is known as the tRNA. Now the tRNA means the transfer RNA. So what's the main function of the tRNA? This tRNA will transfer the amino acid for the ribosome, for the protein synthesis. So it brings the amino acid to the ribosome for the protein synthesis. This is the main function of the tRNA. Now next one is the rRNA. rRNA is the ribosomal RNA. That is the round globular-like structure, and the main function of this is to maintain the makeup of the ribosome. It maintain the structure of the ribosome. So that is the difference between the 3 different types of RNA. Now this one is another part of the genetics. Now what you have to do is you have to know the various terms that are used in the genetics. Now you may come across the gene, locus, alleles, homozygous gene, heterozygous gene. So you have to know definition, or you have to understand what those mean. The gene, just see this one is the chromosome. Now can you see a small light part of A, capital A? So this is known as a gene. So the gene is sort of the DNA. This is the functional part of the DNA that determine the characters of an individual, because this is the gene which is transferring from one generation to another generation. This will play a very important role in determining the character of an individual. This is known as the gene. Now what do you mean by the locus? The gene, the location of the gene, the position of the gene in a chromosome. This position is known as the locus. So jani ra gene gaera bस्छ नि त्यो ठाउँलाई, that is known as the locus. Now next one is known as the alleles. So alleles भनेको के हो भने, so you can see this is the homozygous chromosome. Last time previously भनिसकेछु हैन, homozygous chromosome को के हो? For the two chromosome to be a homozygous chromosome, the position of the centromere should be, you can see the position of the centromere at the same level. Then the position of the gene, which is determining a particular trait. So that position of the gene should be a same locus. Okay, so that's known as a homozygous gene. Now consider the homozygous gene. In this homozygous gene, this one is occupying the identical position. So this is known as the alleles. So this capital A and capital A is known as the alleles. 11 is the alleles. Is it clear, you? Is it clear, this one? Hello?
Yes, ma'am.
सुनेछ क्लियर छ कि रिपिट गरिदिनु पर्छ?
एकचोटि रिपिट गरिदिनु न म्याम.
Okay, homologous chromosome त बुझ्यो हैन? This two hole is known as the homologous chromosome. Now when you have to know about the alleles, now you just see both the gene that is determining a particular trait, same trait, जस्तै यो जीनले eye color दिन्छ भने यो जीनले पनि eye color दिनुपर्छ. Now both the gene which is determining the particular trait are occupying the same position in a chromosome. This A, capital A and A is known as the alleles. 11 is the alleles. बुझ्यो अब?
Yes, ma'am.
Okay. Now next one is the homozygous gene. So the homozygous gene, when the two parental genes are identical for a particular trait, that is known as the homozygous gene. Now you just see over here, this one, the capital A and the capital A, though that is known as a homozygous gene. Now this one is known as a heterozygous. Capital A and a small a is known as a heterozygous. Why this is known as a heterozygous gene? Because this is a pair gene which is denoting the contrasting feature of the same trait. That means if this is determining the tallness, if this is determining the shortness. So those are the contrasting feature of the same trait, that is known as the heterozygous gene. अब alleles भनेको के हो भने capital A and the small a, capital A and the capital A, all are known as alleles. Okay? So we have the alleles over here. There also this is known as the alleles. This is also known as the alleles, and this is known as the alleles. You सबै alleles. Now, what do you mean by the dominant gene? What do you mean by the recessive gene? Now, say in this diagram, we have three different cats, color of the cat. So the color the same sa duita koi. This is the brown one, this is the brown one, and this is known as this. You can see this is the capital B will represent the brown, the small b will represent the gray one. Now see, when you see the genetic makeup of this one, you can see for the genetic makeup, you have to see this one. Capital B is there, capital B is there. This is known as the homozygous or the heterozygous. यसलाई homo भनिन्छ कि hetero भनिन्छ?
Homo.
Now next one, the small b and the small b. This is known as a homo or hetero?
Homo.
Homo, okay. Now what about this one?
Heterozygous.
Heterozygous. Now consider this one. Take two examples. This is the homozygous, this one is the heterozygous. Now which one is the dominant gene in this case? The capital B and the capital B, that is known as the dominant gene. That means the dominant gene will express its characteristics in a homozygous or in a heterozygous state. Jun state ma bhai pani homozygous state ma bhai pani heterozygous state ma bhai pani यसले आफूले express गरिहाल्छ. That is known as the dominant gene. Now, due to the expression of this dominant gene, the cat is having a brown color. Right. Now, next is see the recessive gene. Which is the recessive gene in this example? Which one is the recessive gene?
Gray, small b.
Small b, okay. Now in the case, now the small b la express हुनको लागि के चाहिन्छ? The recessive gene will only express in a homozygous state. That means if both are the recessive gene, then at that condition only this is going to express. That is known as the recessive gene. Now, what about the small b in the case state? In the case of the heterozygous, the small b will act as a carrier. So this is known as the carrier gene. This is the heterozygous recessive gene that is not going to express in the first generation, but this may express in a succeeding generation because this will act as a carrier gene, and it may express in a successive generation, not in a first generation. That is known as the carrier gene. बुझ्यो अब यो carrier gene भनेको के हो अब? Now next is see the phenotype. What do you mean by the phenotype and what do you mean by the genotype? So the phenotype is just see this example over here. The color, जुन हामीले color देख्छौं त्यसलाई नै phenotype भनिन्छ. So the outer appearance, the physical, biochemical, psychological appearance of an individual, which is resulting from the expression of the gene, that is known as the phenotype. Now what do you mean by genotype? A phenotype भनेको tall हो कि short हो कि color कस्तो छ त्यो सबै they all come under the phenotype. Now next one is the genotype. So what do you mean by the genotype? The genotype means the genetic makeup of an individual. Now if you see this one, this cat, so what is the genotype of this cat? This is capital B, and the capital B, that is the genotype. Now due to the expression of this genotype, the cat is having a brown color. That is the phenotype. Now this is all about the various terms which are used in the genetics. Now is it clear? Kisa, do you have any question regarding this one?
No, ma'am.
के सोध्नु पर्ने छ?
No, ma'am. Clear, sir.
Okay, now next one is the Mendel's experiment. Now you know about the Mendel's law, Mendel experiment. Now what he had done is he did an experiment taking a 7 different contrasting character in a pea plant. So he observed the inheritance. He observed the inheritance of the 7 different contrasting character of the pea plant. Now what are those 7 contrasting character of the pea plant? So kind of seed. One, the round one and the wrinkle one. The gray and round, the white and the wrinkle. Next one, in the case of cotyledon, one is the yellow, next one is the green. Next, in the case in the case of the flower, white flower and the violet one. Now the pod, the form full one and constricted one, the yellow one and the green one. Now in the case of the stem, the position of the flower in an axial one, in a terminal one. Now next one, the length of the stem, the long one and the short one. These are the 7 different contrasting character of the pea plant, and what he had done is he observed the inheritance pattern of all those, and at last he found out 3 different law. He stated the 3 different law. So law of segregation, law of independent assortment, and law of dominance. So these are the 3 different laws. Now just see the law of segregation, law of independent assortment, and law of dominance. So the law of segregation, now just see this one. It's the long one, and next one is the short stem. So he did the cross pollination and artificial pollination between the long and the short stem, and what he found is he stated the law of segregation. Now, what does this law of segregation means? Now, see, this is one. This is the one parent. This is the another one, and the small tt for the short one. Now, what he had observed is each individual possesses the 2 genes for a particular trait, capital TT for the tall and small TT for the short one, then out of this capital TT or capital TT, only one of this will get separated and they will transmit it to the next generation. That means their offspring will receive 1 gene from the father, 1 gene from the mother, or 1 from one parent, 1 from another parent. So that is known as the law of segregation or the law of separation. So the 2 gene small TT नै जान्छ मात्रै जान्छ एउटा प्यारेन्ट्सबाट एउटा मात्रै जान्छ भनेको के. So this is known as the law of segregation. And अब pair of the now just say this is the capital one, this is the small one. Now can you observe what will be the phenotype of this plant? What will be the phenotype of this plant?
Tall.
That will be the phenotype. अब यो tall र short mix हुँदैन यसमा. The pair of the in a hybrid form will never get mixed with each other. You mix हुँदैन. So this is known as the law of segregation. You बुझ्यो अब law of segregation? अब law of segregation कुन केसमा fail हुन सक्छ? This will get fail during non-disjunction. अब non-disjunction ma I'll tell you later on what you mean by the non-disjunction. So it may get fail during non-disjunction. That may increase the chromosome number, and we have chromosomal abnormality. That is a separate, different case. Now, next one known as the law of independent assortment. So, for the law of independent assortment, the 2 parents differing from each other in 2 or more points of the contrasting character are taken. So, over here is having a round and the yellow seed. This is the round and yellow seed, and this is the wrinkle and the green seed. भनेपछि त एउटा शेपमा पनि डिफरेन्ट भयो, अर्को कलरमा पनि डिफरेन्ट भयो. So the 2 parents, if the 2 parents differing from each other in 2 or more point of the contrasting character, in that case, if you take that one, then they will inherit one or independent of the another. That means यो यसमा round गयो भने yellow पनि जानुपर्छ भन्ने छैन. The expression of this allele, the expression of round allele will not be affected by the yellow one. So, as a result, we can get anything. We get round, okay. We can get round and green. Round and yellow नै आउनुपर्छ भन्ने छैन के यसमा. That will not affect the expression of another one. अब यसमा पनि के छ? यसमा पनि round and okay, just take this one. We have got the wrinkle and the yellow one. That may appear. So the expression of one allele will not depend upon the expression of another allele. So it will express in any way. It can express in one way, anyway. त्यो बुझ्यो. अब म फेरि भन्दिन्छु. अब यो राउन्ड एक्सप्रेस हुने बित्तिकै त्यसको साथसाथमा येलो एक्सप्रेस हुनुपर्छ भन्ने छैन. यसमा पनि रिंकल एक्सप्रेस हुन्छ भनेपछि त्यो रिंकलको साथसाथमा ग्रीन नै एक्सप्रेस हुनुपर्छ भन्ने छैन कि. So the expression of one allele will not affect the expression of another allele. That is known as the law of independent assortment. They will express as an independent identity or entity. That is law of independent assortment. Is it clear, this one? Buzio? Hello?
Yes, ma'am.
Okay, shall I move on?
Okay, ma'am.
Okay. Now the last Mendel's law is known as the law of dominance. So the law of dominance, for the law of dominance, let us take the 2 flower, different color. One is the purple, next one is the white one. So over here, this purple color, this is the dominant gene. The gene which is going to express the purple one is the dominant one. Let us consider this one. Then this dominant gene, again, the dominant gene is going to express in the first generation. So all the plant in the first with the purple flower, this is known as the law of dominance. So those dominant genes will appear in the first generation, whereas the recessive gene will not appear. And the recessive gene may appear in the next generation. That is known as the law of dominance. बुझ्यो अब यो? Is it clear this one? अब second generation मा हेर त 75% of the flower, those 75% are the purple flower and the 25% is the white flower, because this is the recessive one and this is the dominant one. This is known as the law of dominance. So we have already finished the 3 different law, Mendel's law of dominance, law of independent assortment, and law of and law of dominance of Mendel's law, which we have already learned about this one. Abuzio, do you have any? Hello? One. So this is these are the term which we have already studied. Alleles. About what you have to know is you have to know about the alleles: gene, dominant gene, recessive gene, homozygous, heterozygous, genotype, phenotype, and next one is the 3 different law, Mendel's law. Now the another most important question, that is the pedigree chart. This is also one of the most. So what do you mean by the pedigree chart? What are the symbols that are used in the pedigree chart that might come from the genetics part? So pedigree chart, this is the family history, or this is the genetic history of the family that has been observed over the several generations. So this is a genetic history of over a several generation that is known as the pedigree chart. So this genetic history of a family of over the several generation will be represented. Pedigree chart. Now, why we have to know about the pedigree chart? What's the most important thing of the pedigree chart is the pedigree chart. The use of the pedigree chart. The pedigree chart is mainly used to determine the distribution of the disease in an affected family. Our normal families are disease pedigree chart is not necessary for that one. But if a family is having any genetic disorder, then is very important. So this is very important to determine the distribution of disease in an affected family. That is, we use the pedigree chart. So pedigree chart, I'll give you one example. If a couple want to know their offspring, or if parents want to know the probability of their child suffering from the muscular dystrophy, parents ले आफ्नो बच्चा muscular dystrophy बाट affected हुन्छ कि हुँदैन भनेर जान्नु पर्यो भने what the genetic counselor will do, the genetic counselor will make a pedigree chart of those family, of the father and mother both, and they will try to find out the person which has been affected from the muscular. अब पहिला सुरु कसले भएको थियो muscular dystrophy, त्यो find out गरेर they will calculate the probability of their offspring. They will calculate the chance of their offspring to suffer from the muscular dystrophy. That's why this pedigree chart is very important. The pedigree chart will begin with the person through whom family came in. So the same example I'll take, the muscular dystrophy. If in a father's family, some grandfather is suffering from the muscular dystrophy, then grandfather will be a proband for that one. So that person is known as a proband because he is the first person to be diagnosed with that particular disease. So it will be considered as a proband. And we will use the propositus for male and proposita for female. And this proband will be marked by an arrow. So you can you see this arrow? And this is a particular symbol that will be in the pedigree chart. So if a if a proband is the female one, then this symbol will be used. If a proband is the male one, then this symbol will be used with an arrow. Now these are the various symbols that are used in the pedigree chart, and you have to memorize this all symbol. You symbol yaad garai, because this is What do you mean by pedigree chart? Why we use, what is the importance of the pedigree chart, and what are the various symbols used in the pedigree chart? These are the three most important questions. Now, for the male, we use the square one. For the female, we round one, and neither a female. So for that, we use the diamond shape. Now, if a child is adopted, then we just keep that one in a box. Okay. If someone is pregnant, then a P would be P will write the P in the middle of diamond shape. There is someone dead, then the dead indicated by the cross one. Okay. If someone is affected with that particular disease, then we'll mark whole. Okay. But if someone is a carrier for that trait, then half. Okay. Now you can see both are the carrier, but these are the difference in one is the autosomal. If someone is the carrier for the autosomal disease, just say the half part. If someone is the carrier, of the sex-linked disease, either the X1 or the Y1, then we'll just put a dot. अब X-linked छ भने female मा dot हाल्नु, Y-linked छ भने male मा dot राख्ने हो. So that is will indicate the carrier for the autosomal and the sex-linked disease. Now this is the symbol for the mating. It's the marriage between the relative. आफ्नै नजिककोमा marriage भयो भने we'll just mark like this one. This is the sibling. That means the family is having a one female and the two male. So number of the children, you can write divorce, miscarriage is indicated by this one. Now next one is a dizygotic and monozygotic. Our monozygotic twins are the identical twins, so we have to join this two. But in the case of the dizygotic twins, they are not so identical. So, but they may be a male, that may be a female, or male-female. जे पनि हुन सक्छ. तर monozygotic twins मा both will be either female, both will be either male, because they are being developed from the same zygote. त्यही भएर कि मेल मात्र हुन्छ कि फिमेल मात्र हुन्छ मोनोजाइगोटिक ट्विन्स. कहिले पनि छोरा छोरी त्यसरी हुँदैन है. That is the dizygotic twins. Now no offspring will be marked by this is stop mark. Now this is the male proband, this is the female proband. Okay? So these are the various symbols that are used in the pedigree chart. Is I'll give some information about the mutation. The shortcut one. So mutation may occur in a single gene or in a chromosome. So whether the mutation has been occurred in a gene or in a chromosome, on that basis that is divided into the one: genetic mutation, gene mutation, and the chromosome mutation. Okay? So the mutagen is the agent that is known as a mutagen, and it may be a physical, chemical, or the radiation. Now you just see there are the two types: chromosomal mutation is there, gene mutation is there. Mutation we have these are changes occurring in the structure of the chromosome. Either the structure of the chromosome can be altered, has been altered. कि number increase हुनु पर्यो अथवा decrease हुनु पर्यो कि structure change हुनु पर्यो. That is known as the chromosomal mutation. Now in the next class you'll know about the structural abnormality and the numerical abnormality in the chromosome. Right? The next class and that class will be taught by Robita ma'am. Now just see, last time in previous slide I have mentioned about the non-disjunction. Only what has to be there in the case of meiosis 1 and in the case of the meiosis 2. Just see in the meiosis 1, this pair of the chromosome has to enter into the one daughter, and second meiotic division one pair into normally के हुनु पर्थ्यो, the each cell has to receive the one chromosome हैन. Haploid हुनु पर्थ्यो, यहाँ 2 छ भने haploid हुनु पर्यो. One, another one fuse भयो भने as a trisomy, and this cell will have the single chromosome from another cell, or normal cells fuse. So this cell will have a single chromosome that is known as a monosomy. So this is the main reason. So the chromosome number may get increased, chromosome number may get decreased, and the main reason for this one is the non-disjunction. That process is known as the non-disjunction, the failure of the separation of the cell during mitotic division. That is known as a non-disjunction. Will affect the individual, known as a gene mutation. Okay. Now, before starting or before learning about the particular mutation, you should have some idea about the mitosis and meiosis. Mitosis, meiosis, this is the same thing which you have already learned in the +2 level also. So I'm just going to revise the mitosis and the meiosis in a short way. Okay. So mitosis is the equatorial division, okay, reductional division. Oh. Hello? Mitosis is the equatorial or the reductional division? Hello? Hello? सुनेको छ? Okay. Now that one is the equatorial division. तपाईले टाइप गर्दाखेरि मलाई थाहा हुँदैन कि मैले स्क्रिन शो गरेको हुन्छ नि. Now, so the mitosis is the equatorial division. It has got the prophase, metaphase, anaphase, and telophase, 4 stages. Now, what will be there in the prophase? In the prophase, the chromatin will appear, the chromosome will appear as a thin thread-like structure. A thin thread-like structure that is known as chromatin. Now, in the case of late prophase ma, the thin thread-like structure will get split into the chromatid. My last time bhaneko thiye ni chromatid bhaneko ke ho. So they will get split except at the centromere. Centromere ma baik sub duita ma split huncha. Oh, so this will happen in a late. In the case of the metaphase, all the chromosomes will aligned at equatorial plane. As the metaphase. Now what will happen in the anaphase? There will be the division. The centromere will get divided and the half. The centromere will divide and one sister chromatid will enter into the one cell, will move to the one pole, will move to the another pole. Now in the case of the telophase, this will, you can see the disappearance of the spindle fiber and you can see the half part of the chromatid in one pole and another pole. The nuclear membrane will appear, the nucleolus will form, and this will lose its appearance and finally convert into, or finally turn into the thin thread-like structure. So the chromosome will regain its from turn. So this is all about the mitosis. Okay. Now next is the meiosis. So the meiosis is a quite complicated one. We have to remember the two. The meiosis will be completed in the 2 division, and the meiosis 2. The meiosis 1 is the longer one. It also has the prophase 1, metaphase 1, anaphase 1, and telophase 1. And the prophase 1 will be divided into 5. Oh, diplotin, and dikinase. This is the further step or further division of the prophase 1. Now in the prophase 1, there will be the synapses. In the prophase 1, we have the thread-like structure, thin thread-like structure which has been split into the chromatid. Oh, you can see the chromatid over here, and there will be the synapses of the homologous chromosome. Homologous chromosome or ekarka nazik bhaera aera formed huncha hai prophase 1. And this what will happen? Now next one is next most important event that will occur in the prophase 1 of the meiosis is one is a crossing over. You know about you पनि थाहा छ है synapses of the sister chromatin, then we have the crossing over कुन स्टेजमा हुन्छ Okay, I'll tell you. In the diplotin stage, there will be the crossing over. The genetic material of the one chromosome will get accents with another one. So you can see the accents of this material over here. In the case of the diplotin. Now, after the dikinesis, our dikinesis proceed. Then anaphase 1, then telophase 1, and which is similar to that of the mitosis. Now in the metaphase 1, this already changed. Already you accents भएको chromosomeहरु हुन्छ नि यो already crossing over भइसकेपछि they will align at the equatorial plane. Observe the difference between the mitosis and the meiosis. Mitosis र meiosis मा अघिकोमा के थियो? We have got the division of the centromere. Not be a division, or there will not be a division of the centromere. The centromere is not going to divide over here. So in the telophase 1, we will get the 2 chromosome, we will get another 2 chromosome over here.
Excuse me, ma'am.
Hello?
Ma'am, हजुरको audio कुनै कुनै बेलामा अड्किरहेको छ.
कतिखेर देखियो? अहिले हो? रिसेन्टली हो कि अघि नै देखियो?
अघि एकछिनको लागि भयो थियो, अहिले चाहिँ अलि धेरै बेरको लागि रोकियो.
ओके, मेयोसिस कहाँबाट रोकियो त? मेयोसिस स्टार्टिङबाटै हो मेयोसिसको?
होइन, मेयोसिस र माइटोसिसको डिफरेन्स भन्दै हुनुहुन्छ.
ल म भन्छु फेरि है. सो द मेयोसिस र माइटोसिसको मेन डिफरेन्समा यु हेभ टु सी द एनाफेज 1. एनाफेज 1 मा हेर त प्रिभियसली माइटोसिसको एनाफेजमा के थियो? सेन्टोमियर हेज बीन डिभाइडेड अनि त्यस हाफ क्रोमेटिड. यो क्रोमेटिड हो नि त हैन, मैले भनिसके थिएँ लास्ट टाइम क्रोमेटिड. अब हाफ द क्रोमेटिड विल एन्टर इन्टु द one cell and half will be another cell. So this cell will have got the 4 number of the chromosome as that of the parent. You the same chromosome number भयो. But in the case of the anaphase 1, the centromere is not dividing chromosome. एउटा पोलमा गयो, अर्को half of chromosome अर्को पोलमा गयो. The chromatid over here, there is no any migration of the chromatid. There is the migration of the chromosome. That's why in the case of the telophase 1, this cell will have the half number of chromosome, and this will also have the half number of the chromosome compared to the parent cell. Hello?
Yes, ma'am.
Okay. Now next one is the meiosis 2. Meiosis 2 is similar to that of the mitosis. Already reduction the bhaisakyo hamro chromosome ma haina. So there is no any reduction in the chromosome in the meiosis 2. So the meiosis 2 ma that will start from the prophase to metaphase 2 ma party start garda ni huncha. So we have the metaphase 2, anaphase, telophase 2. So you just see the prophase ma what will happen in the prophase 2? The chromatid. You can see over here this will appear as a chromatid. Now this will form a tetra. This will get duplicated and form the chromatid except as centromere. When you chromo shape of regain गर्छ भनेको. Okay. Now next one is in the case of the metaphase. This will align in the equatorial plane. Now in the case of the anaphase, this anaphase 2 is similar to that of the anaphase of mitosis. Over here, the centromere is dividing. Centromere dividing. So the centromere, the half of a chromatid, the chromatid of, or the half chromatid of the chromosome will enter into the one pole and another will enter into the, and finally we'll have the two different cell, and this will have the same number of chromosome as that of the parent chromosome इन द केस अफ मियोसिस 2 हैन मियोसिस 2 को स्टार्टिङमा जति नै नम्बर अफ क्रोमोसोम थियो त्यति नै डटर सेलमा पनि भयो आई एम नट टकिङ अबाउट द मियोसिस 1 मियोसिस 1 लाई भनेको थिएन है सो मियोसिस 2 मा जति नै क्रोमोसोम थियो इनिशियली त्यसमा पनि सेम नम्बर अफ क्रोमोसोम भयो सो इन दिस वे all together in a combined all together we have the 4 daughter cell consisting of the half of chromosome as that of the parents cell अब यहाँ एउटा सेल थियो भने चारवटा सेल with or 4 daughter cell with half number of chromosome so this is what is happening in the meiosis so this you have to know बुझ्यो अब meiosis र माइटोसिस को डिफरेन्स यो त तिमीलाई थाहा छ त हैन प्लस 2 मा पनि पढिसकेछ now okay this one is the end of the class right today's class so if you have any queries about the pedigree chart symbol or the Mendel's law you can ask me non-disjunction Okay. Non-disjunction मा अघि हेर्दा meiosis 1 meiosis 2 हामीले पढिसक्यौं नि हैन meiosis 1 मा के थियो meiosis 1 मा is prophase 1 metaphase 1 anaphase 1 telophase 1 अब prophase 1 मा पाँचवटा थियो लेप्टोटिन जाइकोटिन पेसाइटिन अब you have to consider all those है now अब anaphase recall गर prophase भयो metaphase भयो metaphase मा all the chromosome align at the equatorial plane म यसमा भन्दिन्छु है एनाफेज मा के थियो यस्तो इक्वेटोरियल प्लेनमा थियो हो मेटाफेज मा इक्वेटोरियल प्लेनमा थियो अब एनाफेज मा के भयो यो क्रोमोसोम सेपरेट भएर जानु पर्ने हो नि त हैन यो क्रोमोसोम सेपरेट भएर हाफ क्रोमोसोम यता जानु पर्ने हाफ क्रोमोसोम यता जानु पर्ने अनि यस्तो हुनु पर्ने तर के हुन्छ भने हाफ क्रोमोसोम सेपरेट नभइकन यो होल क्रोमोसोम यो दुईटै क्रोमोसोम सबै यो सेपरेट नभइकन एउटामा गयो रे सेपरेसनै भएन क्रोमोसोम भनेपछि त यो सेलमा क्रोमोसोम नम्बर हुँदैन that is known as a non-disjunction about this cell has to enter into this has to enter into this one तर यसमा के भयो both the chromosome is entering into a single and whereas other cell is lacking अब यसको फेरि meiosis 2 मा अब यो सेल meiosis 2 कम्प्लिट त भएको छैन माइटोटिक डिभिजन अब माइटोटिक 2 सेकेन्ड meiosis 2 डिभिजन अन्डरगो गर्दाखेरि के हुन्छ देयर विल बी द सेपरेसन अफ द क्रोमाटिन सेपरेसन हुन्छ भनेपछि दुईटा फेरि यहाँ जान्छ दुईटा फेरि यहाँ जान्छ यो दुईटामा त यहाँ त क्रोमोसोमै छैन भनेपछि यो खाली हुन्छ अब is having a 2-1 this is also having a 2 this is having a 0 now if this cell will fuse with the normal one suppose this is the normal if this cell will fuse with the यसमा पनि के हुन्छ तीनटा हुन्छ यसमा के हुन्छ 1 यसमा के हुन्छ 1 so these two are known as monosomy these two will be trisomy when they will get fused with the healthy another cell बुझ्यो अब नन डिसजंक्सन अनि यसरी नै हामीले क्रोमोसोमल एब्नर्मालिटी हुने हो के अब डाउन सिन्ड्रोम हुन्छ डाउन सिन्ड्रोममा के छ ट्राइसोमी 21 क्रोमोसोममा हो 21 नम्बरमा 3 क्रोमोसोम हुन्छ 3 क्रोमोसोम कसरी भयो नन डिसजंक्सनको कारणले भयो सो दिस इज द फर द नन डिसजंक्सन इज इट क्लियर दिस वन बुझ्यो अब ओके अरु के छ सोध्नु पर्ने
पेडिग्री चार्ट युजेस भन्दिनु न चाहिँ
पेडिग्री चार्ट के भन्दिनु भन्न सबै भन्दिनु कि सिम्बल भन्दिनु कि के भन्नु
युजेस
युजेस र इम्पोर्टेन्स अब पेडिग्री चार्टमा मेन युजेस भनेको that will help to determine the distribution how the genetic disease will distribute in an affected family जस्तै कुनै पनि प्यारेन्ट्स छ भने उनीहरुको अफस्प्रिङमा उनीहरुको चाइल्डमा कतिको डिसअर्डर आउन सक्छ जेनेटिक डिसअर्डर कतिको प्रोबेबिलिटी कति छ त्यो क्याल्कुलेट गर्ने नि पेडिग्री चार्टले हो के त्यो पेडिग्री चार्टबाट हामीले क्याल्कुलेट गर्न सकिन्छ अब पेडिग्री भने the pedigree chart is about recording the genetic history of the family over the several generation so मैले अघि एउटा एक्जाम्पल दिइसके नि if the parents wants to know their offspring or if the parents wants to know the probability of their offspring suffering from the muscular dystrophy पनि genetic disease हो अब थाहा पाउन मन छ भने अब के गर्छ जेनेटिक काउन्सिलरले उनीहरुको फ्यामिली हिस्ट्री लिन्छ अब फ्यामिली हिस्ट्रीबाट कसलाई मस्कुलर डिस्ट्रोफी थियो, अब जस्तै ग्रान्डफादरले थियो कि ग्रान्डमदरले थियो कि ग्रान्डफादरले थियो कि त्यो हिस्ट्री लिनु पर्यो। त्यो हिस्ट्री लिएपछि अनि उसले चाहिँ प्रोब्यान्ड, क्यान यु सी द प्रोब्यान्ड, यसरी मार्क गर्छ जुन पर्सनलाई फर्स्टमा मस्कुलर डिस्ट्रोफी लागेको हो उसको फ्यामिली हिस्ट्रीमा, दे विल मार्क एज अ प्रोब्यान्ड अर दे विल कन्सिडर दिस वान एज अ प्रोब्यान्ड। अब त्यहाँबाट उनले प्रोबेबिलिटी काउन्ट गर्छ, प्रोबेबिलिटी क्याल्कुलेट गर्छ। सो दिस इज अबाउट द पेडिग्री चार्ट। यही नै हो इम्पोर्टेन्स पेडिग्री चार्टको, बुझ्यो? हेलो हेलो
बुझ्यो म्याम।
ओके, म यो त मेरो क्लास होइन है, म तिमीलाई एउटा चार्ट चाहिँ देखाइदिन्छु है। छैन यसमा, हेर हो, यो अटोजोमल डोमिनेन्ट इनहेरिटेन्सको यसो। अब यो प्यारेन्ट्स भयो, अब ग्यामेटमा कोलाई कति पर्सेन्ट चिल्ड्रेनहरू एफेक्टेड हुन्छ, कति पर्सेन्ट चिल्ड्रेनहरू हेल्दी हुन्छ। अब यसमा के छ, हाफ अफ द अफस्प्रिङ चाहिँ हेल्दी हुन चान्स हुन्छ, हाफ एफेक्टेड हुन चान्स। यसको मतलब एउटा सिंगल प्रेग्नेन्सीमा 50-50 चान्स भयो कि एफेक्टेड हुने, 50% चान्स एफेक्टेड हुन सक्छ, 50% चान्स प्रोबेबिलिटी नन एफेक्टेड हुन सक्छ। यो यसरी नै क्याल्कुलेट गर्ने हो। अनि त्यो पेडिग्री चार्टबाट हेरेर नै अनि क्याल्कुलेट गरिन्छ हामीले, बुझ्यो अब यो?